Start, sir.
Okay, sir. Okay. Good morning, dear students. Welcome back to another live online webinar class for the subject of zoology conducted by Telangana Residential Educational Institutions Society. Dear students, this is Raj Kumar, PGT in Biology from TSRS&JC Lingampally. Dear students, we have been learning zoology from past 2 days. We have completed 2 chapters so far. If you have not watched all those classes, go to the official site of TRICE and watch those classes and watch this. Okay, today we are going to start a new chapter from zoology second year. Let's see what we are going to learn. Here on the screen, second year zoology, and we are going to learn about excretory products and their elimination. It is the unit B. It is a unit B from your second year zoology. Okay, without taking much of your time, let's get into the class. So what we learn in this chapter called excretory product and their elimination. We have already covered this class, but today we will be learning about the important questions. In this excretory products and elimination, we study about the excretory system. Excretory system is composed of kidneys, ureters and urinary bladder, isn't it? If we, and not only learning about the external structure of the kidneys, we study about internal structure of kidney, which you can see here, the LS of kidney. This is also important for 4 marks question. And we study about the internal structure of the kidney. Okay, kidney has nephrons. Nephrons are structural and functional units of the kidney, isn't it? So we study about the nephrons as well. If we observe the nephron under a microscope, we can see this kind of structure. You can see here. Okay. Kidneys and LS of kidney, the structure of the nephrons and ultrastructure of the nephron. Let's get into the class now. What we are going to learn from today's class. We will be having an explanation on question and answers. We will have a discussion on important questions which may come in the examination for IPE. And we have a discussion on 2 marks question, 4 marks questions and 8 marks questions. We also have some of the NEET questions which are important from this chapter. Okay, we will discuss how the questions are asked in the previous NEET exams. We will have questions as well. Okay, let's get into the concepts. Let's see the first question, that is for 2 marks. What are the column of Bertin? Column of Bertin, where do they exist? Here in the picture, it is already shown between the 2 renal pyramids. There is a structure which is dividing the 2 renal pyramids. That's called column of Bertin. See the answer. In the human kidney, the renal pyramids are separated by the projection of the cortex called the column of Bertin. Okay. Let's see the second question. Distinguish between cortical and juxtamedullary nephrons. Okay. Cortical nephrons, the name itself says that cortical nephrons exist in the cortex region of the kidney. Juxtamedullary, the name itself says these nephrons extends to the medullary region of the kidney. What is the answer? Let's see the answer now. The loop of Henle is too short and extends only very little into the medulla in cortical nephrons. Okay, so their loop of Henle is short in the cortical nephrons, whereas the loop of Henle are very long and run deep into the medulla in juxtamedullary nephrons. So it's all about the length of the loop of Henle. If the loop of Henle is short and if it remains only in the cortical region, it's called cortical nephrons. If the loop of Henle is long enough to get into the medulla region, it's called medullary nephrons. So, okay, so this is the basic difference between cortical and juxtamedullary nephrons. Let's see in the slide which are the cortical and which are the medullary nephrons. With the help of a diagram also we can see here juxtamedullary nephrons, Marfesian capsule, PCT, DCT lies in the cortex region, whereas the loop of Henle extends medullary region, which are called juxtamedullary nephron. Here in the diagram on slide, see the slide, I'm pointing it. So this nephron, which has a long loop of Henle, which is extending to the medullary region, is called as juxtamedullary nephron. Next one is cortical nephrons. They have short Henle's loop exist within cortex, vasa recta are the capillaries surrounding the Henle's loop. Here you can see another nephron which is very short. In them you can find there is a short Henle's loop, and the Henle's loop only present within the cortex region, hence it is called cortical nephron. In juxtamedullary nephron, the Henle's loop is extending in the medullary region of the kidney. Okay, hope you understood this question. Let's go to the third 2 marks question. Define glomerular filtration. So when we learn about the formation of urine, we have studied that there are 3 steps, right? The first step is the glomerular filtration. Let's see what is this. The first step in the formation of urine is the filtration of blood from glomerulus into the lumen of the Bowman's capsule, and this is a passive process called as glomerular filtration. Okay, so glomerulus means the group of blood vessels. Group of blood vessels making a ball-like structure is called as a glomerulus. Glomerulus present in the Bowman's capsule, right? And from the glomerulus, whatever the waste substances are there, nitrogen is waste along with the useful material, all those gets filtered into the Bowman's capsule. Right? This is called glomerular filtration. Let's see the next one, the defined glomerular filtration rate. Out of this glomerular filtration, there will be the filtrate formation. That's called the primary urine. Okay, primary urine. So define glomerular filtration rate. What is this? See, glomerular filtration rate, it is a volume of fluid filtered from the renal glomerular capillaries into the Bowman's capsule per unit of time. Okay, this is called glomerular filtration rate. Let's go to the fifth question, fifth 2 marks question. What is juxtaglomerular apparatus? Okay, so just now I have shown you the picture. There also you can see the juxtaglomerular apparatus. It is the composition of 2 parts of the nephron. Macula densa together with juxtaglomerular cells form the juxtaglomerular apparatus. Okay, macula densa is present in the DCT region. DCT means distal convoluted tubule. Okay, there you can find this macula densa. It's a group of cells with juxtaglomerular cells is called juxtaglomerular apparatus. The next question. Distinguish between the enzymes renin and renin. Okay, these are 2 enzymes which has different functions. Okay, but their spelling is somehow similar, renin and renin. Okay, let's see what are these 2. First of all, let's see about the renin, normal NIN. It is an enzyme secreted by juxtaglomerular cells of the nephron, and this enzyme catalyzes the conversion of angiotensinogen into angiotensin. Angiotensinogen is the inactive form of a angiotensin. Okay, so this inactive angiotensinogen converts into angiotensin with the help of an enzyme called as renin. It is secreted by juxtaglomerular apparatus. Juxtaglomerular apparatus is the combination of macula densa and juxtaglomerular cells. Okay. Let's see what is meant by renin, NNIN. It is a proteolytic enzyme found in the gastric juice of infants. Okay, so it's also called casein, calcium ostracasinate. Okay, it is useful for the digestion of milk protein. Okay, so renin is useful for the digestion of milk protein, whereas renin is useful conversion of angiotensinogen into angiotensin. Okay, let's go to the next 2 marks question. Distinguish between juxtaglomerular cells and macula densa. So these two kinds of cells are located in two different regions. Okay, that's the basic difference between these two. The wall of the efferent arteriole has juxtaglomerular cells. Okay, so these juxtaglomerular cells are present in the wall of the efferent arteriole. They are modified smooth muscles of the efferent arteriole. Whereas a group of modified epithelial cells of the distal convoluted tubule are crowded in the region constituting the macula densa. So basically, juxtaglomerular cells located in the arterioles, okay, afferent arterioles of the blood vessels. But when it comes to macula densa, they are located in the DCT region. DCT means distal convoluted tubule. That's a basic difference between these two. What is meant by the term osmoregulation? Okay, osmoregulation is very important. It is repeated most of the time. Process of maintaining the quantity of water and dissolved salts in balance. This is important. In balance is referred to as osmoregulation. Here, the quantity of water, the amount of solutes should be in balance so that there won't be any endosmosis and exosmosis. Over endosmosis and over exosmosis both are harmful for the body. So the water content and salt content should be balanced in such a way. That's called osmoregulation. Let's see the next question, which is for 4 marks. Okay. Differentiate vertebrates on the basis of nitrogenous waste products they excrete. Okay, giving some examples. So according to the nitrogen waste that they produce, animals are of 3 types. Okay, ureotelic animals, uricotelic animals, and ammonotelic animals. Let's see one by one. Ammonotelism means the elimination of ammonia as the chief nitrogenous waste material is termed as ammonotelism. Where do we see this? In the aquatic animals like fishes, we see this ammonotelism. That means they excrete ammonia. As a nitrogenous waste. Next is ureotelism. The name itself says ureotelism means excreting urea from the body. Okay, their nitrogenous waste is the urea. That's why the term called ureotelism. Ureotelism is exhibited by earthworms, amphibians, and mammals. Okay. Next is uricotelism. The name itself says uric acid. So the elimination of uric acid as the chief nitrogenous waste material is called as uricotelism. Uric acid is excreted by arthropods, reptiles, and birds. Arthropods means the insect family, you know that. Reptiles means lizards and snakes, and birds. These excrete uric acid as their nitrogenous waste. So according to their nitrogenous waste, animals are of 3 types. Let's go to the next question. This is for 4 marks. Explain micturition. The urinating process is called as micturition, basically. Okay. Let's see what happens in the micturition process. Answer, you should write this. Urine formed by the nephrons is ultimately carried to the urinary bladder, where it is stored till a voluntary signal is given by the central nervous system. Okay. So it is it is stored in the urinary bladder. So what happens in this? See the step by step. The signal is initiated by the stretching of the urinary bladder as it gets filled with the urine. In response to the stretch receptors on the walls of the bladder, they send signals to the central nervous system. When the urine is more in the urinary bladder, so what happens is stretch receptors gives the signals to the CNS, that is central nervous system. The CNS passes on motor messages to initiate the contraction of smooth muscles of the bladder and simultaneous relaxation of the urethral sphincter, causing the release of urine. Okay. The process of passing out of urine is called the micturition, and the neural mechanism involved is called the micturition reflex. Okay, so this is what the process called micturition. Hope you have understood. You should remember only 3 steps. The first step is when the urine is filled in the urinary bladder, the stretch receptors which are present on the walls of the urinary bladder, they pass on the stimulus to the central nervous system. Central nervous system, what it does, it initiates the signals for the contraction of smooth muscles in the urinary bladder. Okay, so that the urination, passing of urine, take place. This is called micturition. In the diagram, you can see how the process takes place of the micturition. See, the pons varoli is giving the signal, and this is the urinary bladder. There are stretch receptors on the urinary bladder. They send the signals, and there is an external sphincter and internal sphincter to the urinary bladder. The internal sphincter is involuntary, whereas external sphincter is voluntary in their function. Okay? So this is how the urinary bladder has the connection with the brain, with the pons varoli for the passing of the urine. That's why the urination is under control of us. In the children, urination is not under the control of them because the internal sphincter and external sphincter both are involuntary in them. But as they grow up, the external sphincter becomes voluntary, that is under the control of the control of the person. Okay. Let's see the next question for the 4 marks. Give a brief account of counter current mechanism. Counter current mechanism is seen in the formation of the urine. Okay, so as we have studied in the previous classes, it has 3 steps: glomerular filtration, tubular reabsorption, and tubular secretion. So on the tubular reabsorption and secretion, we see this counter current mechanism. What is this? See this, mammals have the ability to produce concentrated urine. Okay. The Henle's loop and vasa recta play a significant role in this. The flow of renal filtrate in the 2 limbs of Henle's loop is in opposite direction and thus forms a counter current. The name itself says counter means opposite. Current means flow. Okay, so here the flow of the fluids is opposite to each other between the Henle's loop and vasa recta. Okay, since they are opposite to each other, it's called counter mechanism. So since the fluid inside is flowing, it is called current. Okay, counter current mechanism. Okay, let's see, the flow of blood through the 2 limbs of vasa recta is also in a counter current fashion. Okay, the proximity between the Henle's loop and vasa recta, as well as the counter current of renal fluid and blood in them, help in maintaining the increasing molarity towards the medullary interstitium. Okay, transport of substances facilitated by the special arrangement of Henle's loop and vasa recta is called as counter current mechanism. So in the next slide, we will learn in detail about counter current mechanism. How does it take place with the help of a diagram? No problem. See the next point. This mechanism helps to maintain concentration gradient in the medullary interstitium. While you're writing the answer for 4 marks, make note that write in steps so that the invigilator, the person who corrects the paper, he will understand it easily. Okay. don't mix it and write like a paragraph, write like a stepwise. In making it as a steps, you can write. Let's see the 8 marks question, which is the excretion, the formation of urine. Under the excretion, what do we learn? It's a life process, and we learn about excretory system and the biochemical process which is involved in the formation of urine. So under the excretory system, we studied about the organs involved in the excretion, like kidneys, and kidneys have nephrons, which are ultrastructures of the kidneys, ureterus, urinary bladder, and urethra. We have studied about this in the excretory system. Under the biochemical process, we study about the formation of urine. This is the 8 marks question and most important. Under the formation of urine, there are 3 steps: glomerular filtration, tubular reabsorption, and tubular secretion. Let's see the first, formation of urine, which is the long answer type question, 8 marks. For this, you should write like this: glomerular filtration. Make a subheading as glomerular filtration and see what happens in this. It is the filtration of blood from glomerulus to the Bowman's capsule. This here in the structure, you can see the glomerulus, which is the network of the blood vessels, and this is the Bowman's capsule. Together with both of them, it is called malfeasant body. Okay, it is the filtration of blood from glomerulus to Bowman's capsule, and it is a passive process. Hydrostatic pressure in glomerulus. So it is because of the hydrostatic pressure which is present in the glomerulus and also the vessels. In the glomerulus, we can see 60 mmHg, and glomerular colloidal osmotic pressure is 32 mmHg. Okay. Exerted by the non-filtered plasma proteins of the blood. And Bowman's capsule also have the hydrostatic pressure, which is 18 mmHg. So total 60 minus 32 plus 18, 60 mmHg is exerted by the blood, and around 50 mmHg is exerted by the Bowman's capsule. So both are working opposite together, and that's why we are minusing 60, minusing this 32 and 18 in the 60. So out of net pressure is around 10 mmHg, which is working over the glomerular filtration. This causes the filtration of blood through 3-layered filtrate. Okay, so blood goes to the glomerulus, and from glomerulus through fenestrations, which are the small minute holes, it is filtered. Okay, from fenestration it goes to the podocytes of the podocytes are the specialized cells in the Bowman's capsule. Okay, so capsule, hence the process is called as ultrafiltration. See here, glomerulus, from the glomerulus, whatever the filtrate is formed, it is called as the primary urine. What is present in this primary urine? Primary urine contains carbohydrates, albumins, globulins, water, nutrients, minerals, etc. Okay, except proteins, all constituents are present in the primary urine. Since it has useful products also, these useful products are reabsorbed in the next step. 1100 to 1200 ml filtered per minute. So imagine the filtration rate of the glomerulus. Glomerular filtrate is 125 ml per minute. So it is around glomerulus can filter around 180 liters of we can produce 180 liters of urine in a day. Okay, so but only we excrete 1.5 liters, isn't it? All the rest of the primary urine which is formed will be reabsorbed. Okay, normal excretion is just 1.5 liters per day, and 99% of the liquid is reabsorbed. Let's see selective reabsorption and secretion. About 85% of the primary urine is reabsorbed by proximal convoluted tubule, which is PCT, and a descending loop of Henle, which is obligatory and which is the mandatory. Okay. Rest of the reabsorption is regulated based on necessities. Okay, so we have studied that there are 3 steps. First step is glomerular filtration, where the primary urine is formed, which in very large amount with useful substances. So these useful substances which are filtered must be reabsorbed. So that is called selective reabsorption. It is called selective reabsorption. Why? Because the reabsorption is so selective. Only the useful materials, useful products are reabsorbed. Okay, the water, carbohydrates, proteins, and whichever minerals. These are the reabsorbed materials, reabsorbed things. But creatinine, phosphate, urea, these are the nitrogenous phase which has to be eliminated. So these excretory products are not reabsorbed. Only the useful ions, useful minerals, and the water is reabsorbed. Substances, useful substances are reabsorbed. According to the reabsorption of the substances, these substances are divided into 3 types. First is high threshold substances. High threshold substances means the mostly and effectively reabsorbed substances. See, essential and effectively reabsorbed substances are called high threshold substances. These are glucose, amino acids, vitamins, and some salts. Low threshold substances are there. These are absorbed very little amounts. Example, urea and uric acid. Under the necessity of for the ionic balance, these urea and uric acid are reabsorbed only in very little amounts. A threshold substances which are not at all reabsorbed. They are actual excretory products like creatinine. Okay? Creatinine is the actual excretory product. Let's see reabsorption and secretion. PCT. What are the things reabsorbed and what are the things which are secreted? Here in the green color, you can see the secreted substances. Okay, creatinine, antibiotics, diuretics, and uric acid are the secreted to the renal tubule. That which goes into the renal tubule are called as secreted substances. And in the red color, the substances are shown which are reabsorbed: NaCl, K, and H2O, which is the water, HCO3, glucose, and amino acids are reabsorbed. Okay? This PCT, which is proximal convoluted tubule, is lined by simple cuboidal epithelium with brush border. 70 to 80% of electrolytes and water are reabsorbed. Sodium is actively transported into the cortical interstitium fluid with transfer of positive charge drive the passive transport of Cl minus and glucose and amino acids. Okay. PCT maintains pH and ionic balance by selective secretion of hydrogen ions and ammonia into filtrate, and whether the reabsorption of reabsorption of copper and HCO3 from it. Okay. So here in the picture, you can see the green color substances are secreting and red color substances are reabsorbed. Okay, movement of water occurs by the osmosis process. Let's see reabsorption and secretion in the Henle's loop. In the Henle's loop, you can see there is a descending limb and there is an ascending limb. Okay, this is called as Henle's loop, which is in the U-shaped structure. Reabsorption is very minimum in the Henle's loop. Descending limb is permeable to water and impermeable to electrolytes. Hence, reabsorption continues and filtrate concentration gets increased gradually as it moves towards the downwards. As it moves downwards, what are the things reabsorbed? NaCl, K plus, and H2O. Water is getting out of the Henle's loop. Since the water is going out and some minerals are getting outside, the filtrate which is present gets more concentrated as it moves downwards to the medullary region. Okay. Ascending limb proximal segment is thin, which in which NaCl diffuses out into the interstitial fluid passively. Okay, here you can see the NaCl is moving outside. Distal segment is thick, in which the NaCl is actively pumped out. The ascending limb 20, ascending limb, then ascending limb, you can see NaCl is moving outside. Since the NaCl is moving outside, the solute concentration is going outside. That is the reason as the filtrate moves upwards, its concentration gets decreased because of the removal of some salts from filtrate. Okay, it is impermeable to water. Okay, water does impermeable to water, does the filtrate becomes progressively more dilute as it moves to the cortex region. So the filtrate in the descending limb, as it moves downwards, the concentration of it gets decreased due to the removal of water from it. And as it moves upwards, the concentration of the filtrate gets again increased, again decreased. Why it decreases? Because of the removal of the salts from it. Okay, let's see what happens in the DCT region, which is the distal convoluted tubule. This is not PCT, actually DCT. In the distal convoluted tubule, cells are shorter and lack microvilli, which indicates the less reabsorption. Okay, so it has no villi structure. That's why reabsorption is very less here, but there is a selective reabsorption. Enzyme driven reabsorption takes place in the DCT whenever it is necessary. Conditional or facultative reabsorption of water takes place here. Conditional means that which is done by certain signals. Okay. Reabsorption of water is dependent upon several conditions and it is regulated by ADH. It is antidiuretic hormone for maintenance of pH and sodium-potassium balance. So on this DCT, distal convoluted tubule, whenever there is a low water, low hydrated, whenever the dehydration is more in a person, if he is losing more fluids from the body, he needs more reabsorption. When a person needs more reabsorption, the ADH is secreted by the pituitary gland. ADH is nothing but antidiuretic hormone. It is also called as vasopressin. This vasopressin, what it does, it acts on the DCT, distal convoluted tubule, and it increases the reabsorption of the water. When the water is more reabsorbed, it mixes with the blood and the person gets hydrated. Okay, he will not be dehydrated fast. To protect from dehydration, this act is performed. Okay. Let's see the next, reabsorption and secretion of the collecting duct. This is the last part of the nephron. Okay, so let's see what happens here. It's a long tube that carries the filtrate through the middle area to the renal pelvis. Here in this distal collecting duct, water reabsorbed slightly in this segment. It allows the passage of small amounts of urea to the medullary interstitium to keep up its osmolarity. Okay? The urine in the collecting duct is hypertonic to the plasma of the blood. So finally, you can see the urinary secretion is equal to glomerular filtration minus tubular reabsorption plus tubular secretion. This is the equation by which you can understand what is the actual urine which is removed. So whatever the primary urine formed by glomerular filtration is 99% reabsorbed. Okay, so after the reabsorption, whatever the substances secreted by the by these tubes, they are the actual excreting substances. Okay, that is the final net urine which is formed in the kidneys. Let's see the mechanism of concentration of the filtrate. Henle's loop and vasa recta plays a significant role in producing concentrated urine. Okay. So how does it produce? Here you learn about the countercurrent mechanism also, which is the most important concept for your IPE and also for the NEET exam. Okay. Let's see. The flow of renal filtrate into limbs of loop of Henle is in opposite direction, isn't it? The flow of blood through the two limbs of vasa recta is also in the counter current pattern. Counter current means, as I said, counter means that which is opposite to each other. The current means the flow. So the flow in the vasa recta and the tubule is opposite to each other. Hence, it is called as counter current mechanism. This countercurrent mechanism helps in maintaining increasing osmolarity towards in medulla interstitium, that is 300 osmolarity in the cortex to the 1200 osmolarity in the medulla. This gradient is mainly formed by transport of NaCl and urea. Okay. NaCl, sodium chloride, enters a descending limb, makes it more concentrate. NaCl goes out in the ascending limb, making it dilute. Okay. The movement of sodium chloride in and out makes the filtrate more concentrated and dilute. Okay, so the concentration and dilute condition is seen in the limbs. This is about the formation of urine, which is a long answer type question, also important for your NEET exam. Now let's see some of the NEET questions. Let's practice some NEET questions now. So far we have learned about 2 marks questions, 4 marks questions, and also the 8 marks question. We had explanation regarding all those, and we have seen some pictures also. If you have any more doubts regarding these questions, you can consult your teacher. Your subject teacher is there in your school. You can ask him doubts, or you can come up to me. I will clarify your doubts, no problem. And let's practice now some of the NEET questions regarding this lesson. See, we will go one by one. First, there are around 15 to 16 questions which are previously came in the examination, and let's see what they are. First question, see on the slide. A decrease in blood pressure or blood volume will not cause the release of what it does not cause the release of atrial natriuretic factor, which is released by the heart. Okay, whenever there is an increase in the blood pressure, this atrial natriuretic factor is released. Okay, so it is released by the heart. Remember, this atrial natriuretic factor is the answer for the first question. Second question, pick the correct statement. Ascending limb of loop of Henle is impermeable to water, isn't it? Just now I have told you in the slide, right? Descending limb is permeable and ascending limb is impermeable. From descending limb, water is moving outside, and from the ascending limb, what is happening? Water does not move outside because it is impermeable. Only the salts moves outside. Okay. So for the second question, A is right answer. Let's go to the next question. A part of nephron involved in active reabsorption of sodium is, which is the part of nephron which is involved in the active reabsorption of, it is the Bowman's capsule. Okay. Next, typically human urine is acidic. Why it is it is acidic? Because hydrogen ions are actively secreted into the filtrate. So what is mean by pH? Actually, acidity. Whenever the hydrogen ions are more in number, that will be acidic, isn't it? So that's why the A is the right answer for the fourth question. Let's see the fifth question. Which of the following causes an increase in sodium reabsorption in the distal convoluted tubule? Reabsorption is increased. Why reabsorption? It is the increase in the aldosterone levels. Okay. Whenever there is more aldosterone, which is produced by adrenal gland. Okay. So the reabsorption will be increased. Let's see the sixth question. Maximum amount of electrolytes and water, about 70 to 80% from the glomerular filtrate is reabsorbed. In which part of the nephron? So just now I told you the nephron has several structures like glomerulus, which is glomerulus, Bowman's capsule, proximal convoluted tubule, which is the immediate part of the Bowman's capsule, and distal convoluted tubule and Henle's loop. So, in all these structures, the proximal convoluted tubule, which is also called as PCT, right? PCT, proximal convoluted tubule, has more reabsorption. Around 70 to 80% of the reabsorption takes place in this proximal convoluted tubule. Okay, let's go to the next question. A fall in glomerular filtration rate activates what it activates. Whenever the filtration is decreased, they release renin. Okay, renin. Juxta glomerular cells, they release the renin. What is the story of renin? Let me tell you. Renin, what it does, it converts the angiotensinogen into angiotensin. Okay, angiotensin. This angiotensin is an enzyme. It converts the angiotensin angiotensin 1 to the angiotensin 2. So these angiotensin 2 stimulates the adrenal cortex. Okay, this adrenal cortex, whenever it is stimulated by the angiotensin 2, which is the enzyme, this cortex of the adrenal gland starts releasing a hormone called as aldosterone. So aldosterone is important for the reabsorption of sodium ions and the water. Okay, so whenever the glomerular filtration is less, so this renin and aldosterone activation system is activated. Hope you have understood this. When I taught you the lesson, I taught this in detail. Okay, what are the changes take place in the aldosterone renin system? Go to that lesson and see that. Let's see the eighth question. This is a common characteristic observed in both human beings and the frogs, adult frog. What is the common one? The frogs, adult frogs and the human beings both excrete only urea. Okay, they are excreting urea in the form of a nitrogenous waste. Okay, since adult frogs and human beings are ureotelic animals, this is the common characteristic that we see. What about the tadpoles of the frog? In the tadpoles, there is a release of ammonia. Okay, so they release ammonia since they live in the water. Okay, so the animals which live in the water, which if their habitat is water, they excrete mostly ammonia. Okay, for the secretion of ammonia, for the release of ammonia, they need a lot of water. Okay, to conserve the water, the human beings and all the terrestrial animals excrete nitrogenous waste in the form of urea. Urea also requires some water to excrete. Okay, and uric acid in flying animals like birds, they excrete uric acid. So it is for the protection of water. That means to prevent water loss, they excrete uric acid. Okay. Uric acid is formed in the form of crystals and they release in the form of that. That's why birds do not urinate. So they excrete uric acid as their excretory product. Let's go to the next question. This is not a part of a renal pyramid. Which one of the following is not the part of the renal pyramid? He is asking. Loop of Henle, collecting ducts, and peritubular capillaries. These are not the loop, part of renal pyramids. Convoluted tubules. These are the part of renal pyramids. In the second slide, I have shown you the difference between cortical and medullary nephrons, isn't it? There we have seen half of the part of the nephron remains in the cortex, and other part is extended to the medullary region. This medullary region contains the renal pyramids. Okay, that's why convoluted tubules, PCT, DCT, and Henle's loop. These come under the part of renal pyramid. Okay. Next, this among us the following correctly explains the function of specific part of the human nephron. Which one? See the last option, which is D. Podocytes. Podocytes. I told you the podocytes are existing in the Bowman's capsule. Okay, what do? They are helpful for the filtration of blood, isn't it? What they do, they create minute spaces for the filtration of blood into the Bowman's capsule. Okay? That is a function of the podocytes. So regarding 10th question, the D is the right answer. You can see the arrowheads. I kept the arrowheads for the right answers. Let's see the next question. Accumulation of urea and other waste substances in the blood is called, what is it called? Hemodialysis or cystitis and uremia and urethris? The answer is uremia. Okay, uremia is the accumulation of urea and other substances in the blood. Next, presence of RBCs in urine. If the red blood cells are seen in the urine, what is its indication? The answer is hematuria. Hematuria is the condition in which the RBCs are found in the urine. Let's go to the next question. Presence of dash in urine indicates the diabetes mellitus. Diabetes mellitus is a sugar disease, isn't it? So under these conditions, under the condition of this sugar disease, they excrete glucose and ketone bodies in their urine. These both are seen. That's why the answer regarding 13th question is the D. Let's see the next question. If a person undergoes a prolonged fasting, then his urine will be found to contain higher levels of. So a person is having a more fasting. That means if he is not eating for a long time, in them we can find the ketone bodies. The answer is ketone. For the 14th question, the answer is ketone. Let us see the next question. Diuresis. What is meant by? The name itself says diuresis means a dilute condition. That means when it is so dilute, we start losing more amounts of water from the body. That's called uresis. So which one is the right answer for this? C is the right answer. C, what C says, body starts losing excessive amounts of fluids. So this condition of losing the excessive amounts of fluids from the body is called as diuresis. Okay. Let us see the last question, that is the 16th question. Presence of RBCs in urine. Just now we have seen. This is actually repeated one. The answer is hematuria. Hematology means the study of blood, isn't it? Hematology. So hema, the name itself, the initial name itself indicates that it is related to the blood, right? So if person found blood in the urine, it is a condition called hematuria. Okay, hematuria. Hope you have understood the 16th question as well. So these are the different questions I have made for you. So hope you have understood the class. So thank you, and thank you for watching. If you have any more doubts, go to first listen to the class. Excretory products and elimination. The classes are available in our TRICE YouTube channel. You can go to there. There you can find, if you type excretory products and elimination, you find all the classes. Watch those classes and watch this revision class. In this revision class, we have only covered the question and answers QA session for the examination, mainly focusing on the IPE exam. Okay. If you want to prepare it for the NEET exam and other exams, watch the complete classes which are earlier made. Okay, and I would like to tell to all students that whatever you see in this webinar classes, take a notebook and write it on a paper. Okay, practice on a paper so that you can remember it for a long time. So for the 8 marks questions, you have to draw the diagrams also. That's why practice diagrams. Okay, with all labelings. And for 2 marks question and 4 marks question, write as much as possible. Which suppose if the 2 marks question is asked, for 2 marks question only 1 or 2 lines are enough. You need not to write a whole answer like 5-6 lines. Some people write long answer for a short answer question. Should not do that. Okay, so if it is 2 marks question, write in 2 or 3 lines. If it is 4 marks question, write in 4 or 5 lines. That's all. If it is 8 marks question, you can elaborate the answer as much as possible. Yeah, I wish you all the best, dear students. Thank you. Hope you have understood. Sir, the class is completed. Thank you.
Okay.